I look like Bin Laden, huh?
You see, today, two people, they occupied with the weapon and they go to the market in Baghdad and explode themselves.
Well, those suicide
38
bombers
people died.
in Baghdad?
Yeah,
Right? Oh, that's too bad. Why are they still doing
these
that?
radical Muslims. They think if they do,
They go to heaven.
exactly. They are stupid.
You're right.
I'm serious.
Well, I believe you.
There is
I
no
agree.
way God, he say, kill yourself and kill other people and go to heaven. There is no way.
It doesn't make sense.
Anyway, I will see you.
Okay. We're doing histology, right? We started that thing? Really? What's today? Thursday? Okay. I'll get it right one of these days. Membranes and cavities. So that's oh, you're right. That was Tuesday. I remember. Oh, yeah, we were getting ready to, okay. Ah, it's coming back to me. We still have a minute or so. Let's see. Get out of there. Wait. Membranes and cavities. Right there. Okay. There we go. I think we're on. I think we're on point there. Let's see here. You can get up there. That looks good. Let me fix the lights. Don't need that. Okay. Time to get started. Today is Thursday, January 21st, 2021. And it is 11 o'clock. You guys are biology 1160 section 2, right? I think that's right. And last time we were finishing up the anatomical terminology, we got to membranes and cavities. And we were talking about the body cavities. And now membranes that kind of define some of these. And so I wanted you to understand and appreciate the different types of membranes there are, because we're going to talk about a lot of these. And so what are they? What are the different types of membranes? Well, there are mucous membranes, or they're collectively referred to as mucosae, or singular would be a mucosa. That's one. Put an E on the end, that's plural. What are they? Well, mucous membranes or mucosae, they line internal cavities or organs or passageways. So you have like a hollow structure, a hollow organ of some sort. It could be trachea, esophagus, stomach, intestines, or whatever. It's hollow. It's always going to be lined with a mucous membrane. And usually the mucosae have specific names, you know, for that specific organ, all right? And so, Jesus, what do they do? Well, being a mucous membrane, they secrete mucus. Now look at the spelling of mucus, M-U-C-U-S. Mucus, that's the secretion. But the adjective that's describing the membrane, M-U-C-O-U-S. There's a why is it like, I don't know. But the slippery lubricant that's secreted by mucosae is M-U-C-U-S, mucus. I don't know. That's how it is. I.e., these are specific examples of mucous membranes or mucosae, like the oral mucosa. That's the mucous membrane lining the oral cavity, intestinal mucosa, vaginal mucosa. Of course, the list goes on. Any hollow structures be lined with a mucosa, okay? Now, there's another type of membrane, but these are more at body cavities. These people coming in late. They're called serous membranes, or in the same context of mucosae, they're collectively referred to as serosae, or a singular serosa. So a serosa would be a serous membrane. What does serous membranes do, and how are they different than mucous membranes? Well, it's basically where you find them. They do pretty much the same thing. Their product is a little bit different, but serous membranes line body cavities. We identified some body cavities, right? Remember those? So you're never going to just have a body cavity that's doesn't have any kind of just random tissue adjacent to it. It's always going to be coated or lined with a serous membrane. And likewise, you're never just going to have a body cavity, or rarely, you're going to have a body cavity with nothing in it. You know, there's going to be something in it. So the structures that are occupying that cavity are also covered with a serous membrane. So you got serous membranes lining the cavity, and then on the surface of the organs that are in those cavities. And they produce a fluid just like mucous membranes do. But typically the fluid or the product that's secreted is just referred to as serous fluid. Okay, and it tends to be less viscous, like runnier, waterier. Is that a word? Watery, more water-like, I guess. It's not as thick and tacky and sticky as mucus. But anyway, it serves the same purpose, that it lubricates and moistens the structures in the cavity. Okay. Examples, i.e., of serous membranes or serosae. Well, there's two broad categories of serous membranes. Those that are associated with the body cavity, and then those that are associated with the organs in that body cavity. Those that are lining the cavities are collectively referred to as parietal membranes, or parietal serosae, okay? Those that are on the organs are visceral serosae, or just visceral membranes. Of course, the word visceral means organs, right? Like viscera is organs, so visceral means of the organs. So you have different types of visceral membranes and different types of serous membranes, but collectively they're all different types of parietal, different types of visceral, but they're all collectively serous membranes producing serous fluid. And so here I talk about some specific types of serosae in the thoracic body cavity. You know, there's body cavities up there we defined, and of course organs in them, right? And here I talk about first the parietal pleura. Now you're putting some words together. So we define parietal, we define pleura. So parietal pleura, what is that? Parietal, remember, that's the serous membrane that lines the cavity. So which cavity? Well, pleura. Remember I told you a pleura, anytime you think, pleura literally means the side, but anytime you hear pleura, you think of the lungs. So the parietal pleura would be the serous membranes that line and define the pleural cavities, okay? And so if you look at this, let me get my laser here. If you look at this graphic right here, and this little inset, these are both the right and left pleural cavities, of course, containing the lungs. And you see it zoomed in right here. There's the surface of the lung. There's the pleural cavity itself. But then this green line right there, that's the serous membrane that's outlining the pleural cavity. And so that would be specifically the parietal pleura. And it's there, it forms a nice slick surface for the surface of the lung to rub up against, okay? So there you go. Of course, you have different types. Now, on the surface of the lung itself, there's another serous membrane. This would be the visceral pleura. So of course, the key word here is visceral. Remember, it means visceral of the organ versus parietal, which is of the cavity. So the visceral pleura is on the surface of the lung in the cavity. So here's the lung tissue itself, and you can kind of see, is that purple or something? Like a little purple membrane on the surface of the lung. Of course, that's just the artist's rendition, whatever, just to make it stand out. But the parietal pleura is the green line. The visceral pleura is like, I guess it's purple. And then the cavity or the space itself is just, between the two, all right? So you got these two slick, slimy membranes facing one another. And of course, it allows for the lung to move with minimal friction, right? And then, of course, also in the thoracic body cavity, of course, between the two pleural cavities, you have the pericardial cavity. And so same sort of thing. The parietal pericardium, that's the parietal pericardium. Of course, the membrane is basically defining the cavity of the heart, right? So that's the serous membrane surrounding the outside of the heart. And of course, you're going to have the space, the cavity itself. And then on the surface of the heart, the serous membrane, the visceral pericardium. So again, visceral pericardium, parietal pericardium against one another. Sometimes it's called pericardial fluid. Pericardial fluid is just the serous fluid produced by these two membranes and allows the heart to beat with minimal friction, okay? Some other specific serosae in the abdominal pelvic body cavity. The parietal peritoneum, which a parietal again is for the cavity, but peritoneum, we haven't talked about that. I don't think we talked about this just yet. But peritoneum is the serous membrane that here I have lines most of the abdominal pelvic body cavity. This is a sagittal section through the torso lateral view from the left aspect. So you're looking from the left side in. So you actually see in the right side of the body. And you can see This is the abdominal pelvic body cavity. And this blue line, this light blue line that's kind of going around everywhere, except right here. It kind of dips down into the pelvic cavity, but it kind of goes over the top of the bladder there. That's a big giant serous membrane. It's called the peritoneum. The word peritoneum means to stretch way around. It goes around. So it lines almost the entire abdominal pelvic body cavity. It dips down into the pelvic cavity, but as you can see, it does not include everything that's in the pelvic cavity. So there are infraperitoneal structures, things that are under the peritoneum. But this big, long peritoneal, big, long serous membrane that's stretching way around, that's the parietal peritoneum. And then obviously inside that space, there's all kinds of organs, all right? And the space itself is defined as the peritoneal cavity. It's formed from the parietal peritoneum, okay? And so if you think about it, it's the peritoneal cavity is actually inside the abdominal pelvic body cavity. Does that make sense? It's kind of weird, you know. Are they the same? Well, kind of, sort of, not really. The analogy I always give for the relationship between the peritoneal cavity and the abdominal pelvic body cavity is kind of like, imagine the parietal peritoneum is a balloon, like a balloon that's just inflated with air, you know. Everybody has, you know, played with a balloon before, right? It's blown up, you get a balloon for your birthday, whatever. The latex, the rubber of the balloon would be the parietal peritoneum, okay? The hollow part inside the balloon where the air is would be the peritoneal cavity, all right? But now take the balloon and put it in a box. The box would represent the abdominal pelvic body cavity. So it's like a cavity within a cavity. See how that works? So your abdominal pelvic body cavity basically would just be everything removed and nothing in there. But lining that cavity, you have this serous membrane, okay? And so it's in there and it produces serous fluid, et cetera, et cetera. And we usually identify structures in the spaces within it as peritoneal, within the peritoneum, all right? So peritoneal cavity, parietal peritoneum, and of course the visceral peritoneia would be the serous membranes on the surface of the organs that are in there. So if you look, these are sections like through the liver and parts of the intestine, et cetera. You see the light blue that's covering on the outside, that would be all visceral peritoneum, okay? Now certain serous membranes that would be defined as visceral peritoneia have special names, but I'm going to spare you those in this class. But you can see a relationship between the visceral peritoneum and the parietal peritoneum. Can you kind of see how they're connected? You got these little like junctions, like little bridges that kind of occur between the visceral peritoneum and the parietal peritoneum. Those are collectively referred to as mesenteries. And so here I have mesenteries, which are folds of serosae connecting the parietal and visceral peritoneum together. Kind of like they anchor the two. It's like a liaison between the two. And some of the mesenteries have special names, like this one right here, between, it looks like the dorsal body wall and the stomach. This is lesser omentum. And then there's a greater omentum and the mesentery proper. They're all mesenteries. Basically what it is these mesenteries, they help anchor and hold the organs in place, and also act as pathways or like bridges for blood vessels and nerves to run. So you're going to obviously have blood vessels going to the intestines and the stomach and so on and so forth. They're going to be contained within these mesenteries, and you'll see them. And so, but they're all functional histological structures that produce serous fluid, et cetera, et cetera. So it helps keep all the organs moist and lubricated and holds everything in place. You probably have seen mesentery if you ever eviscerated an animal, like you gutted an animal, cleaned a catfish or a deer or whatever, or just remove the entrails of a chicken or a duck or something. You ever seen between the intestines, there's this kind of clearish, kind of almost like thin cellophane looking tissue, okay? That's the mesentery. The mesentery kind of it's connected to the entrails, but at the same time, it segues to the body cavity. It kind of holds it all into place. There you go. They're all serous membranes. Now, there's a term since we've learned that, I wanted to introduce this term to you if you've never heard of it before. Retroperitoneal. Has something to do with the peritoneum, right? But retro means like behind or posterior to. And so look at this view. This is a horizontal section through the torso at about the level of the second lumbar vertebra. And so it's a superior view looking down. This is the dorsal surface, the ventral surface. And you're viewing into this gray space is the peritoneal cavity. And you can see some organs that are surrounded by visceral peritoneum. And then you can see some little mesentery right there that's kind of a bridge between the mesentery, between the visceral peritoneum, and then of course parietal peritoneum here in the background. This is dorsal mesentery. And of course you have ventral mesentery and so forth. But anyway, these organs are contained in here, but not everything is. Because if you look right here, there's structures and organs that are not in the peritoneal cavity. Classic examples are the kidneys. The position of the kidneys is identified as retroperitoneal because they are posterior to or dorsal to the peritoneal cavity. This is just another view. You have all you're looking into the peritoneal cavity right here. And this golden yellow looking tissue right here in the background is the parietal peritoneum. But you can see over here on this side, it's been removed. And so you can see structures that are retroperitoneally, this retroperitoneal position. These are structures that are behind the peritoneum. And so in other words, you can't really see the kidneys and the abdominal aorta and the posterior vena cava and ureters and so forth unless you remove the peritoneum. And so that's what this refers to. And then also remember that the peritoneum, it comes down and it dips down into the pelvic cavity a little bit, but it covers over the top of the bladder and the uterus and so forth if you had one. And so those structures would be infraperitoneal, so they're underneath it. And so they're not in the peritoneal cavity. And there's a reason for that. It's if you think about it, what's going on in the peritoneal cavity? It's kind of like it's sealed off and kept away from these structures that are behind it or outside of it or underneath it. Well, that's chiefly your digestive system that's in the peritoneal cavity. Of course, your digestive system is where you're acquiring nutrients and things that you want to keep, right? Well, outside of that is stuff that you don't want to keep. What's this? That's basically your body's septic system, right? It's where you're getting rid of toxic waste. And so you really, it's kind of like you don't want to put your kitchen next to the outhouse, right? And so they're separated, so you keep, there's other boundaries that help separate the nitrogenous waste from the digestive system in here. So this just kind of like helps compartmentalize and keeps everything separate. Okay, I think that's it. Look, the end. And so that's all your anatomical terminology. And so next, we have to move on to histology. I think this must be the first class, I think it is, that I've started this. But so good with the anatomical terminology. A lot of that's pretty easy, but I'm telling you, don't blow it off. Don't ignore it. You know, make sure that you're real comfortable with all that stuff, because from this point forward, it's going to be assumed that you know all that stuff already. All right, and because I'm going to use it throughout the course. All right, so no questions about any of that stuff. Okay, well, this is our second PowerPoint. Histology. Remember histology, we defined it when we were defining the divisions of anatomy. And I think I said it was the study of the form and function of tissues. We're studying tissues, and that's what we're starting this week, at least in my laboratory, studying tissues, right? You know, to study tissues, of course, you're taking sections through organs, which are part of organ systems, which are a part of the organism. And remember, there's cells that make up tissues, so on and so forth. So the first thing I want to do, again, kind of revisit this a little bit, is define what a tissue is. So what is a tissue? A tissue, at least here's how I define it. It's a pretty good definition. Defined as a collection of cells with similar form and function, which are specialized for a particular activity. Okay, they're doing something, you know. So not all tissues do the same thing, you know. Your body has certain needs, and of course, you get certain tissues, you know, doing that. Another characteristic of a tissue. Here, I tell you that cells of a particular type, you know, of a particular tissue tend to have the same embryonic origins. Okay, so well, if you think about it, really, I mean, you can trace all cells back to the single cell, but you're going to see that there's all kinds of tissues, and we like to group them and categorize them and label them. And how do you decide on how to make these categories? How do you make these categories? Four main types of tissues, and there's subcategories and so on and so forth. A lot of that's based on where those tissues are derived. If they have the same developmental or embryonic origin, we kind of group them together like that. And so, speaking of embryonic origins, I have a series of pictures or graphics, or I don't know what you call these, kind of showing you where it all starts. What is this? Can you recognize what this is? It's remember we talked about embryology. Embryology, we define it as gametogenesis, fertilization, and prenatal development. Of course, this is representing what right here? This is fertilization, right? Fertilization is the union of gametes. This is where life starts. It's the union of gametes, when you have a fertilized egg. And of course, the fertilized egg is no longer an egg anymore. It's a zygote, a single-celled organism. It's a zygote, fertilized egg. And what happens in the zygote, you know, you basically, your female's egg has a nucleus, but it only has half the number of chromosomes it needs. And so it's not fully human yet. And so really the only thing the sperm cell does is restores that chromosome number. And so it's basically just chromosome with a tail. It swims and it supplies that other set. I gotta turn on my laser again. Set of chromosomes. And so you really have like 2 nuclei at the beginning of fertilization. We call them pronuclei because this is the nucleus of the egg, and of course this is the nucleus of the sperm cell, and of course each one of them has half the number of chromosomes. So if this human, you know, different animals have different number of chromosomes. So if this is human, this pronucleus would have 23 chromosomes, and this pronucleus would have 23 chromosomes. So 23 and 23 together make 46. So 23 pairs of chromosomes is 46 chromosomes total. And that's true. All your cells, with the exception of the sex cells, have 46 chromosomes with 23 pairs. This, it says 2 pronuclei within 24 hours. Okay, that's what develops within 24 hours of fertilization. So the female is inseminated, all right? And if the egg is there and the sperm are, you know, mixed together, this is gonna happen. You're gonna have a zygote with pronuclei within the first 24 hours. So then what? Well, once the pronuclei unite and you have a fully formed zygote, it's gonna start to undergo mitosis. It's gonna start dividing. And then this is the beginning of the early cleavage stages. We call it cleavage because the cell cleaves, it kind of cuts into 2, it splits into 2. And we have a name, so you have the zygote, you start with the zygote, and the next stage of development is the 2-cell stage. It's not very complicated, 2 cells. So there's 1 cell and it splits into 2. And like you might figure, those 2 follow suit, they divide again. And so you go from a 2-cell stage to a 4-cell stage. This is about 48 hours later, or about 48 hours after semination. So about 2 days, you have a 2-cell stage. This is the second cleavage stage, all right? The cells here aren't destined to become anything in particular other than more cells. These early cleavage stages, the cells have not differentiated yet. So, you know, you can't like pick out one and say, oh, this one's going to be the brain and this one's going to be skeletal muscle or whatever. No, actually, if you separated one of the cells at one of these points, it would just continue to divide. As a matter of fact, that's exactly how identical twins are formed. For some reason or another, one of these cells becomes detached and it just keeps growing. Because all these cells are clones of one another. They're genetically identical with one another. So if you separate one, you got clones. And so that's how you would produce identical twins, okay? So there's no differentiation at this point yet. And of course, here, day 2, 48 hours, you're still in the fallopian tube. The fertilization takes place normally in the fallopian tube or the oviduct. And this journey is, of course, going to be the early cleavage stage. Almost all the cleavage stages occur within the fallopian tube. Of course, these 4 divide again. And so you actually have an 8-cell stage, a little bit closer to the uterus, but not quite. And then, of course, you would have 8, and then 16, 32, 64, so on and so forth. But ideally, or theoretically, I guess you could say. But beyond 8 cells, it's difficult to count them and know. So there's a lot of guesswork. And so, so 16 to 32, maybe up to 64 cell stages, we don't really identify them as, you know, a 16 cell stage or 32 cell stage. We identified as a cluster of cells beyond 8 cell, which is anywhere from like 16 to 32 or 16 to 64 cell stage. We just call it collectively the morula, okay? This is anywhere from like 3 to 4 days later. You're about at this point, you're about halfway down the fallopian tube, okay? So what's that? 48, 72, wait, 23, 70, Oh, how many hours is that? A lot. It's down 3 or 4 days. Each 4, that's almost 100 hours. Anyway, 72 to 90 something hours. Anyway, 3 or 4 days. You have a morula. The word morula means mulberry, because I guess to early microscopists, you look at this, yeah, it just looks like a little mulberry. But it's more than just looking like a mulberry. What's the key thing here is that these cells, again, they're undifferentiated. You can remove one right now and it'll still just keep on dividing. And notice they get a little bit smaller. They get a little bit smaller, a little bit smaller, a little bit smaller. And, but what's happening in a morula, it's just a cluster of cells, and it is a solid ball of cells. That's the key word, solid. That there's no hollow spaces or anything forming inside. Okay, so it's just real primitive. All vertebrates go through the exact same stages, just like this. Okay, and so you can study, you know, these early primitive stages in just any animal, really. But at about day 5 in humans, and most mammals, is just like this. At about day 5, well, the timing would be a little bit different, but they still have the same stage. The morula develops into a blastocyst. And that's the key thing. You go from morula to blastocyst. What makes a blastocyst is that it has a hollow center. It's no longer a solid ball of cells. So you're witnessing something that's very different, okay? The cells are starting to become differentiated. They're behaving differently. It's just not a solid ball of cells anymore. You have this hollow center, all right? You're right at the edge of the uterus. You're almost entering the uterine cavity, okay? Implantation occurs within the next 24 or 48 hours. So about a week after insemination, that's when implantation would take place. I say about day 7. Is that the blastocyst, you can see it just is developed a little bit further. You know, the cells get a little bit smaller. You still have this cavity, what they call a blastocyst cavity. It's what's going to actually attach to the endometrium inside the female's uterine cavity. And different portions of the blastocyst were going to go on to form different parts of the embryo. This center mass right here, what do they call it right here? I can't, it's covered up. They call it the animal pole. Here they call it the embryonic pole. In other words, this is the end that's actually going to form the embryo. And then these other cells out here are going to go on and contribute to the extra embryonic membranes, like the amnion and so on and so forth. So it's going to develop in here. So, but this is when attachment takes place. So it's about a week, you know, later. Once implantation occurs, then that's when all hell breaks loose and things start really changing up. So here I tell you, after implantation, cells of the blastocyst begin to differentiate. There's 3 distinct embryonic germ layers that develop. Sometimes the germ layers are referred to as primary tissues, because remember, you know, cells make up tissues, et cetera, et cetera. These tissues now have become basically programmed to become a particular type of a tissue in the adult. So their developmental trajectory is laid down, all right? And once this happens, we identify, you know, these 3 embryonic tissues. Actually, the first 2 developing at the same time together. There's first ectoderm. Ecto means like on the outside, right? And derm means like skin. So like an outer skin, I guess you can kind of think of it. Well, the ectoderm here, I tell you, is the superficial most layer. It contributes to the integumentary nervous systems eventually. And you can kind of see how I have it pointed out right here, this outer layer right here. You kind of tell it kind of looks like a blastocyst. It's kind of modified a little bit. You're going to see this, the hollow center, and then this outer, these tissues here. All this is occurring kind of buried in the endometrium. And then, of course, it's going to start growing out from that. But first is the ectoderm. But when the ectoderm forms, simultaneously you have a second tissue that's endoderm, which literally means like inner skin, inner lining, inner like membrane kind of thing. That's the deepest layer. That's around here on the inside. It contributes to the digestive system and other internal structures. That makes sense because you look, it's the deepest, it's inside here. See this hollow part right there? That's actually formed by the endoderm. And that hollow space right there where my pointer is located is the same hollow space that's in your mouth. The same hollow space that's in your esophagus and your stomach, all the way to your anus. There it is. This is your developing GI tract right there. And of course, this is what it looks like very, very early up in development right here. All right. And then the middle layer, which is actually, you would think they would develop in order, but they don't. The middle layer actually develops last. It's called mesoderm. I tell you, it's the intermediate layer. Remember that word intermediate? It's the intermediate layer. You can kind of see it on both sides here. Here it kind of, it's coming out. It's kind of growing between the endoderm and ectoderm. And here that shows you a little bit further on this side. They usually develop at the same rate on each side, but it grows around both sides, and it kind of separates the endoderm from the ectoderm. And then as development continues, the mesoderm goes on to contribute to all the connective tissue of the body and all the muscle of your body. And that makes sense too, if you think about it. You got these three layers: ectoderm, mesoderm, endoderm. Ectoderm, skin, on the outside. The other thing that kind of seems weird is the nervous system. We'll see how that happens. But basically your skin on the outside, your connective tissue and muscle in the middle, and then all your guts in the deepest layer. Does that make sense? And then, if you, cut through your body, you would find the exact same three layers like that, basically. And so there you go, ectoderm, mesoderm, endoderm, the three embryonic germ layers of the three primary tissues, okay? Now, those three primary tissues, they all differentiate into, different types of adult tissues. So we have 4 classes of adult tissues that develop from these three embryonic germ layers. First, epithelial tissue, or sometimes it's collectively referred to as epithelium. It develops primarily from ectoderm and endoderm. So ectoderm and endoderm both produce their different forms of epithelial tissue. All the connective tissue from your body comes exclusively from mesoderm. So mesoderm goes on to form connective tissue and all your muscle. All your muscle comes from mesoderm too. There's mesoderm, you can see we're going to see that there's 2 different types of mesoderm. There's mesoderm that's going to go on to form connective tissue, then there's mesoderm that's going to go on to form muscle, okay? But you can trace their developmental history back to mesoderm, that middle tissue. And then nervous tissue, this is really weird. Nerve tissue, it comes exclusively from ectoderm. And when we study the nervous system, we'll see how that works, okay? So, what are we going to do now? Well, we want to talk about the tissues, all right? Remember I said there's 4 different types of adult tissues, and we'll talk about them in order like that. Start with epithelial tissue, and then go to connective tissue, and then nerve tissue, and muscle tissue, or muscle the nerve. I think that's the order I use. But what makes epithelial tissue? So I'll give you characteristics and so on and so forth of the classes, and then we'll talk about the different types. So what are some characteristics of epithelial tissue? Well, if you look at it, I got a couple of different forms here, just real basic looking forms. Epithelial tissue classically has closely packed cells. If you look at it, all the cells, if it's 1 cell layer thick or 2 or more cell layers thick, the cells that make up the tissue are jam-packed, closely packed together. So there's not a lot of extracellular space or anything like that. That makes sense if you're just pure cells, there's going to be no or very little extracellular space. And that's very characteristic, very typical of epithelial tissue. And due to the fact that there's no extracellular space, there's not going to be any other structures in there. So epithelial tissue tends to be avascular. Okay, when you're looking at sections of organs and you're looking at some epithelial tissue, you're not going to see blood vessels or anything in it. So that's why avascular means with no blood vessels. So a section of it, you wouldn't expect to see blood vessels or anything like that. It is living tissue, and it's going to require nutrients and produce waste products and so forth. And so it's going to need to communicate with the circulatory system. So here I tell you that the vascular supply from adjacent connective tissue is what services the epithelial tissue. The material is transported via diffusion. So in other words, you have blood capillaries next to epithelial tissue, and material to and from the circulatory system just has to kind of like diffuse through the tissue to the epithelial tissue, okay? Now, another characteristic of epithelial tissue is that it always has two distinct surfaces, okay? It seems kind of weird, but this is the only tissue that does this, and all epithelial tissue does this. First, it has an apical surface up here at the top, or it's known as a free surface. So it's always going to be exposed to nothingness. It's always going to be exposed to the outside world, or the hollow space of an organ, or the hollow space of a body cavity or something, okay? And so here I tell you, it's exposed to a cavity or a lumen or an external surface. The lumen, see that word lumen, L-U-M-E-N? I talked about this in lab for those of you that have had me for lab already. Or maybe I've mentioned it in here. But anyway, a lumen is just the hollow portion of an organ. All hollow organs have a lumen, all right, that space. And maybe I mentioned the word lumen when I was talking about, remember the directional terms, deep and superficial, okay? The deepest layer of a hollow organ would be that adjacent to the lumen. And then the most superficial tissues would be out towards the outside, okay? So anyway, lumen, hollow part. So apical surface. So if you have an apical surface, what do you call the surface that's opposite that? Well, that's the basal surface down here. So this is the basal surface here, and then way down here, basal surface here. Here I tell you, the basal surface is always deep to the apical surface and adjacent to a basement membrane. The basement membrane, that's a buzzword for you, a specific feature that's unique to epithelial tissue. So all epithelial tissues possess a basement membrane. Well, what is the basement membrane? Well, the basement membrane functionally separates the epithelial tissue from adjacent tissue. Like if you have a basement, nobody in Louisiana has a basement, because you can't dig more than like 6 inches before you hit water, right? So, that's why we bury people above ground and so on and so forth. But anyway, up north, everybody has a basement, right? And of course, everybody knows basement's underneath the house. And so you kind of think of the epithelial tissue as the house sitting on the basement, right? So it's underneath it. The basement membrane, always on the basal surface. That's why the basal surface is called the basal surface, because it's adjacent to the basement membrane. And the basement membrane, like I said, it functionally kind of holds the epithelial tissue in place, all right? And, so it acts as an attachment point, site and support for the epithelial tissue, all right? Now, another characteristic, epithelial tissue is highly mitotic, usually. Not always, but most of the time, it's considered to be a mitotically active tissue. Well, what does that mean? Well, if something is mitotic, it means it's undergoing mitosis regularly. Remember the early cleavage stages? One splitting into two, and then those split and they divide. That's mitosis. Mitosis is a type of nuclear division, or we call it karyokinesis in biology. It's basically where cells are dividing. Now, in the cleavage stages, that's for growth, right? You go from a one cell organism all the way up to 100 trillion cells, all right? And so cells divide for different reasons. So growth, reproduction, repair, or whatever. And so if you have a tissue that's undergoing mitosis regularly, it's renewing itself, okay? So it's renewing itself quite often. And the mitotically active cells aren't just anywhere, they are going to be confined to the basal-most cells, or the cells that are adjacent to the basement membrane. Okay, so these cells down here will be the ones undergoing mitosis. And they divide and they grow toward the apical surface. So in other words, you can think about this, the basal cells would be the youngest cells, and the older or more mature cells would be the more apical cells, or the ones closer to the surface. Okay, does that make sense? All right, now, there's two primary types of epithelial tissue, okay? So now we're getting into categories and subcategories, right? First, you have epithelial tissue that's just simply known as membrane epithelium. It's because this membrane epithelium makes membranes. You know, you got serous membranes, mucous membranes, et cetera, et cetera, et cetera. Well, membrane epitheliums are always covering and lining something, just like the membranes I defined earlier, all right? Structurally, the membrane epithelium consists of sheets of cells that cover external surface or line the internal surface of something, okay? So what other kind of epithelial tissue could there be? Well, there's the membrane epithelium, and then there's glandular epithelium. So what's glandular? What's a gland? You know, a gland is something that makes some kind of secretion. It secretes something. There's all kinds of glands. Everybody knows salivary glands, thyroid glands, this kind of gland, whatever kind of gland. Any and all glands of your body are epithelial tissue, or it's glandular epithelium. So that's why I tell you right here, glandular epithelium are all the glands of your body. All glands are epithelial tissue. And likewise, all, well, I don't want to say that. I was going to say all the membranes of your body are epithelial tissues. That's not 100% true. That the word membrane is kind of used loosely. It's kind of kicked around. But anyway, your glandular epithelium consists of glandular cells. So what does that mean, glandular cells? Well, glandular cells produce a secretion, right? That's their job. The cells may occur individually or in organized clusters. And most of the time when you think of a gland, you know, like your thymus gland, thyroid gland, or pituitary gland, or pineal gland, you can see it. It's a multicellular structure, okay? But there are glands that are only one cell big, okay? And if they're individual, they'd be unicellular glands, and we'll talk about those, all right? So here I tell you, again, functionally what they do, you know, secretion, it's the production and release of a fluid substance by a cell. That's a secretion. Examples of secretions would be, you know, well, what do they make? Well, it could be mucus, you know, coming from mucus-producing glands, or mucosae, right? Sweat would be a secretion. Enzymes, hormones, anything, any kind of thing that's secreted would be a secretion. Of course, they all come from glandular epithelium. All right. So, and we're going to talk about different types of membrane epithelium and different types of glandular epithelium. And so here we're going to start with different types or classes of membrane epithelium. So here, classification of membrane epithelium. There's two. The classification, you know, we have all these classification schemes in biology. There's always a scheme. You know, there's always a protocol to follow. You know, this are the rules. This has to be like this, et cetera, et cetera. We define our criteria, all right? And so in classifying membrane epithelium, there's two basic criteria that we use. First, the arrangement of the cells in membrane epithelium. It's always going to be one way or the other. It's either going to be simple or stratified. And we use this term. So almost all the names of the membrane epithelium have the word simple or stratified in them, almost all of them. And so if you read the word simple, it tells you something. Simple epithelial tissue consists of tissue that's only one cell layer thick. That's what makes it simple. It's only one cell layer thick. What if it's not one cell layer thick? What if it's two or more cell layers thick? Well, if it's layered, we don't say layered, we say stratified. So something is layered, it occurs in layers, like one layer, or you can say it occurs in strata, or a strata is plural, singular would be a stratum. So one layer would be a stratum, many layers would be strata. And so if you have tissue that has strata, it is stratified. So in other words, it's two or more cell layers thick. It's a stratified epithelium. That's what you see right here. This is an example of some epithelial tissue. It's 3 cells deep. 3 cells, that's more than one, right? So this would be stratified. Of course, this is only 1 cell layer thick, so it'd be simple. So we always do that. How many cell layers thick is it? And what are the shapes of the cells? Now, the cells, when you're classifying membrane epithelium and you're looking at the cell shapes, you always want to look at the most mature cells. And so that's pretty easy on simple epithelium. All the cells on simple epithelium make contact with the basement membrane, so those would all have the same shape, because those are the most mature cells. There's nothing other than that. But in a stratified epithelium, the most mature cells are found where? I already told you this. Where do you find the most mature cells in stratified epithelium? Yeah, is it on the basal surface or the apical surface?
Apical.
On the apical surface. So you look at the apical most cells, you look at the shape of them, and they're almost always will fall into 3 categories, 1 of 3 shapes, okay? Don't look at the basal cells. The basal cells are not mature yet, and so they don't have their adult or fully formed mature shape yet. You look at the mature shape, and 1 of the 3 shapes would be right here. They'd either be squamous, cuboidal, or columnar. So all membrane epithelium is based on their cell layers and cell shapes, and they have 1 of these 3 shapes. The word squamous literally means scale or scale-like or scaly, and like a fish scale is flat, right? And so if you look and you see these apical most cells are really thin and flat, those are squamous cells. And so squamous cells can be in 1 layer where they would be simple, or they can be in 2 or more cell layers where they would be stratified. So simple squamous versus stratified squamous, okay? And likewise, this next shape, cuboidal, like cuboidal means like a cube, right? What's a cube? Well, a cube, I guess geometrically would be a structure that is just as tall as it is wide. And that's basically what these cells are. They're not like perfect little blocks, you know, like those stacking cubes or those little, what do you call little blocks the kids play with, the little letters and things on it? What are they called? Alphabet blocks? Yeah. When I think cubes, I was thinking alphabet blocks. And those are almost perfect little cubes, you know? But these cells aren't perfect little cubes. However, they're just about as tall as they are wide. And another characteristic of cuboidal cells is they have a nice round or spherical, almost perfectly round spherical nucleus right in the center. Look at this. See, this is, of course, these are artist renditions over here, but this is actually a photomicrograph. And you can see 1, 2, 3 cuboidal cells here. This is the 1 cell, and this round thing in the center is its nucleus. Here's another cell right there and its nucleus and so forth. But look how nice and round that cell, the nucleus is in the center of the cell. And that's, you know, that's pretty characteristic of cuboidal cells. Will look like that, whether it's 1 cell layer thick or more than 1 cell layer thick. So you can have cuboidal cells that are simple cuboidal or stratified cuboidal, whatever. And then this third cell shape is called columnar. You think columnar, that means like a column. You know, you think what a column is. A column is something that is tall and skinny, right? And yes, columnar cells are cells that are taller than they are wide. Okay, they're not always perfect little columns, but if you look at it overall, they're like tall and skinny, or at least taller than they are wide. Here's the basement membrane here on this photomicrograph, and you can see how they're stretching up. This is kind of weird. I don't like how the artist did this, because he or she, I don't know who drew this, but they put some of the nuclei way up here. That's you're never going to see that. All the nuclei, or most of the nuclei anyway, are basally located, and the nuclei take on the overall shape of the cell. So the nuclei tend to be oblong as well, kind of stretched out kind of like a football. So kind of that oval shape, maybe not round, but kind of oval shape and basally located. Now some of them kind of look staggered. Maybe that's what they try to do, like that here. But that's a little bit overboard there. This needs to come down just a little bit more. So, but you never have the nuclei way up here by the surface. They're going to be closer down here next to the basement membrane. Okay. Anyway, 3 cell shapes, 2 cell layers, or 2 different types, either single, simple, or stratified. Okay. Now, with that in mind, I'm going to this slide. I just list all the different types, and we're going to look at these. I just wanted you to have at least one page where you can just see everything. Okay. But we're going to look at all these, because I just want you to understand how these are all grouped together and why they're grouped together the way they are. So you got 2 main categories of membrane epithelium. There's 8 altogether, basically. And the 2 categories are based on whether they are simple or stratified. Simple is just these are all simple epithelium. It's all 1 cell layer thick. So again, these guys, and then of course these all here are all 2 or more cell layers thick. So they're all stratified. And so you have 1 cell layer thick of flat cells, 1 cell layer thick of cube-shaped cells, 1 cell layer thick of column-shaped cells. Say that real fast. And then I throw you a curveball here. So you get simple squamous, simple cuboidal, simple columnar, and then this pseudo-stratified columnar. What the hell is that pseudo-stratified? You got stratified down here, but up here, this one says pseudo-stratified columnar. You got a columnar. This is simple columnar. This is pseudo-stratified. What's going on? What does the prefix pseudo mean? Pseudo. You ever heard that word like pseudo, like pseudopodia? What's pseudopodia? Completely tangent. It means false feet, like the false feet on an amoeba. They call pseudopodia. It's not really a foot. It's kind of like a foot. Well, pseudo-stratified means falsely stratified. If you are saying you are falsely stratified, you're saying what? I'm not really 2 or more cell layers thick, right? Isn't that what you're saying? Well, if you're not 2 or more cell layers thick, then how thick are you? What's simple mean? 1 cell layer thick. So this is a simple epithelium. It's only 1 cell layer thick, but we call it pseudostratified. Why do you do that? Why don't you? Well, you're going to see that this is a columnar epithelium. It's basically cells that are taller than they are wide, but it kind of is the illusion that it's stratified, but it's really not. And we'll see how that is. Then these last 4, there's 8 altogether. These last 4 are basically just stratified versions, where you got stratified squamous, stratified cuboidal, stratified columnar. Then here's your 4th curveball. It doesn't really follow suit, right? It says transitional. So there's a transitional epithelium. If something is transitional, what does that mean? That it's transitional. I'm transitional. If you're transitional, what can you do? You can change, or people can be transitional, things can be transitional. What's changing here? Well, you can't change how thick you are. So transitional is a stratified epithelium. So it's going to be 2 or more cell layers deep. But what's changing, what's undergoing a transition, is the shape. Okay, and we'll see how that is when we look at this type of tissue. It's the shapes of the cells undergo a transition. So sometimes they might look squamous, and sometimes they might look cuboidal. And we'll see why that is. All right, so here they are in all kind of one-stop shop right here, so you can see everything. And then to present each of the 8 types of epithelial tissues, what I've done is I've hijacked this table from your book, this is from your textbook, and it just kind of does everything for you. And so this is what I would want you to study, and I want you to understand what's going on here from this table. This is from the histology chapter. Now, this is from a long time ago. I don't know what edition this was in, but I don't know if it's still in chapter 5. It might be in a different chapter now, but whatever chapter your histology chapter is in your current edition. Or you could just look at this. I mean, it'd be the same thing, same kind of information, but basically what this table does is it profiles all the tissues. It shows you a photomicrograph of the tissue, an example like what it should look like underneath the microscope. And then over here, it actually shows you an artist's rendition of that photograph. And sometimes, sometimes when studying things in biology, looking at pictures obviously helps. You look at it to understand, what's going on. You look at a picture of it. But sometimes drawings, especially things that are abstract, if an artist drawing or illustration, really aids in understanding what's going on, because then artist rendition kind of exaggerates some details so you can understand, like, oh, okay, yeah, I can see, where the cells start and stop and so on and so forth. So all the tissues are going to have this on this table: photomicrograph, artist rendition, and this little inset right here kind of gives you an idea of where you could find that tissue. It's not confined to the little inset there, but sometimes your author kind of used some weird examples, and I would have probably done it a little bit differently. But anyway, that's just what that is. Now, and then underneath, you got some text just kind of describing what's going on. There's 3 main things. There's the microscopic appearance, of course, it's, it tells you what it's looking like. The representative location, like where you'd find it. And then its function, basically, what it does as a tissue. And so that's what you would need to concentrate on, all the tissues, what it looks like, where you'd find it, and what it does. Okay, and so I'm just going to go through the tissues like that, and I'm going to do that for all the tissues. Okay, so what are we going to start with? Well, the simple squamous epithelium, and just really, if a lot of these names of the tissues are pretty descriptive. If you think about simple squamous epithelium, 1 cell layer thick of these flat cells. And sure enough, that's what you see right here. You see that here, okay, you kind of see here's a lumen of some sort or some kind of space. And then you see that these cells right here, they're facing that outer surface. And it's kind of hard to tell, but like the artist rendition, you can see, oh yeah, I can see this is kind of like a series of just these flat cells lining the outside of this organ. And what that was. But there you go. This is a membrane right here, one cell layer thick of flat cells, simple squamous epithelium. So there you go. Here they tell you, I always say this too in lab, that they shape kind of like fried eggs. If you can imagine a fried egg, where this is the egg yolk, and then the cytoplasm would be like the white of the egg, or kind of like, see, there's the yolk in the middle and the cytoplasm on either side. So there's a bunch of fried eggs laid end to end. That's just kind of, or whatever it looks like to you, There you go. Where do you find this stuff? Well, if you think about this tissue, these are all membrane epithelium, so you're making some kind of membrane somewhere. Now, this membrane isn't going to be very structural or very tough or heavy duty if it's only one cell layer thick of, you can say, flat cells. So it's going to be kind of delicate. So there's some representative locations where you would invest in a very thin, delicate tissue like this. And the first representative location that they offer, it says the air sacs or the alveoli of the lungs. Okay, think about this. In your lungs, what's your lungs for? It's for respiration. What's respiration? Well, that's the exchange for respiratory gases, where you get oxygen for carbon dioxide. Well, you actually get oxygen from the atmospheric air, and you got to get those oxygen molecules into your bloodstream. Okay, how does that happen? Well, in your lungs, you got little cavities of air where that oxygen is going to be collected, and then it's going to diffuse into your bloodstream. And so oxygen molecules getting into your blood, you don't want to make those air sacs real thick. You want it to be easy for the oxygen molecules to go through that membrane, all right? You want to go through the air sac into the blood. And so you can't make it any thinner than one cell layer thick of flat cells, and that's why the alveoli of the lungs are made out of simple squamous epithelium, right? And then it goes on to tell you glomerulus of the kidneys and some other things. Well, what is that? The glomerulus of the kidneys. Some of my lab students, I kind of talked about this already, but what the glomerulus of the kidney is a blood capillary. And so it doesn't have to be just the glomerulus, but all capillaries are made out. Doesn't matter if they have a special name like glomerulus or whatever, but all capillaries are made out of simple squamous epithelium, one cell layer thick of flat cells. Okay, for this exact same reason. You want it to be thin, because what do capillaries do? Capillaries are the exchange vessels. That's where you're going to get material leaving the circulatory system and going into your tissues, and vice versa, from the tissues into the blood, and from the blood into the tissues. And so one cell layer thick of flat cells allows for the passage of material like nutrients and waste products through that wall, and oxygen and carbon dioxide through that wall. So all capillaries are made out of simple squamous epithelium. And then capillaries, or they segue to other blood vessels, right? So your blood vessels are obviously connected to capillaries. And so if you follow the blood vessels to capillaries or from capillaries or whatever, you'd basically end back up in your heart and so on and so forth. So you'd be in the circulatory system. And so that lining of the capillary continues and is continuous with the inner lining of all your blood vessels. Okay, so you got simple squamous epithelium, and it actually continues inside the lining of all your blood vessels. And so in an artery or in a vein, the inner lining is going to be the same stuff that forms the capillaries. So simple squamous epithelium, and that's true. So think about this, lining all blood vessels. It doesn't matter if it's an artery, arterial, venule, whatever, or even the hollow chambers of the heart. It's always going to be lined with simple squamous epithelium. And we have a name for this. The inner lining of all your blood vessels is called the endothelium. I saw the word right here. Look, endothelium. See right there? Endothelium is the lining of all your blood vessels, and it's simple squamous epithelium. It's the same stuff that forms capillaries. Now, of course, all your blood vessels are connected to your heart, right? In your heart, there's a hollow organ, chambers inside the heart, and it's connected to the blood vessels. The hollow part of the hollow chambers in the heart, where the blood is continuous with the hollow portions inside your blood vessels, right? The luma of the blood vessels. Well, it's also simple squamous epithelium lining the inside chambers of the heart. And so you just call it endothelium inside the heart. I guess you could, but we don't. We don't call it endothelium. Once you get inside the heart, it's called endocardium. 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 Do they have? I don't think they have that up here. Anyway, I'm just kind of giving it to you. So capillaries, endothelium, endocardium, alveoli. Anywhere else? Well, we talked about these, and this is why I talked about this. There's method to my madness, why I talk about certain things in the order I do. Look, serous membranes, and then list a bunch of organs. Doesn't matter. All serous membranes, if it's visceral or parietal, it doesn't matter. Here they list certain organs, but we can just jump to the chase and say, well, just visceral membranes. Okay, if it's on the intestines or on the stomach, it doesn't really matter. It's still going to be the same stuff. All serous membranes are formed with simple squamous epithelium. Okay, and that's what you're actually looking at right here. See this little inset, I don't know if you can see, but there's a little red ring on the outside. This is the stomach, if you haven't figured that out. That's the stomach, this is the duodenum, and this is the duodenal-jejunal flexure that's segueing to the jejunum, the second segment of the small intestine. And they show you just like a little red ring right there, but it'll be covering the entire outside of the stomach and all the intestine. That's where your serous membrane is. And that's what this is right here. This is a serous membrane right here on the surface, like on the outer surface of an intestine, on the outer surface of the stomach or whatever. You're going to have just this thin layer of simple squamous epithelium, and it's going to be secreting serous fluid. So it keeps the surface nice and slippery, right? It doesn't have to be real thick. And that's generally what happens in membrane epithelium, where you need some extra fortification. It makes it thicker. And so you get some extra protection there. But if you're in areas of the body that, well, there's not going to be a lot of trauma and you got other things protecting it or whatever, it can be simple. It can be once a layer thick. So there you go. So anywhere else? And it talks about other membranes. Okay, but that's pretty much the same. To me, in my mind, it's all superfluous to just read out all that extra stuff. And so its function, well, it just kind of falls in place. If you think about what it looks like and where it's found, and like I explained, again, you can figure out what its function is. You know, it's real thin, it allows for the rapid diffusion or the passage of material from one side of the membrane to the next, et cetera. And of course, it lubricates, with its fluid. And, it wouldn't be, because you're going to have all these other functions that you'll see. But that's just primarily what simple squamous would do. It's thin, basically secretion and diffusion. Those are the two main things, okay? I think that's it. Now, do we get out at 12:20? Okay. I'm going to stop here because it's going to take more than 5 minutes to do the next one. And then we're going to kind of want to get into a roll. And I don't want to stop myself. You won't complain. I don't think you're going to complain. Any objections to stopping 5 minutes early? No? Anybody out there? No, hope not. So anyway, we're going to quit here. And then Tuesday, we'll pick up. Y'all remember this. You tell me, we're going to talk Tuesday. We're going to start on simple cuboidal. Okay. All right. Well, Today's what, Thursday? So you guys are looking at the weekend? Have a good weekend and be good. And if you can't be good at it. Okay? All right. Adios. No questions, huh? All right. See y'all next week.